for sample collection as well. For example, if you're collecting blood, then it should be a recommended tube for that particular assay or application, because uh, if the blood is hemolyzed, then the hemolyzed blood can get you the degraded DNA, and the sequencing will not be good enough for your application. Some applications we are forced to sequence degraded DNA, that is, for example, the microscope slide for a tumor. But there are ways to get data from that as well, but it becomes more and more expensive if you go towards the degraded DNA. So first thing you should understand that DNA comes from the sample. It could be from any source. It could be microbe, it could be a viral genome, it could be a human genome, or any animal or plant genome. First of all, so the next generation sequencing scope is both in humans and non-human samples. Second concept that you should know for sequencing is extraction and purification. So DNA extraction from the cell, I will not go into the detail, but this is very important part, because this is where you start getting into the sensitive steps of the sequencing. So once you have extracted your DNA and purified it, you need to also, one thing they're missing here, but it also comes in extraction and purification, is your quantification of the DNA. So quantification of DNA is very important because we have one slide in our company that says garbage in, garbage out. So whatever you will put in the sequencer, you get the similar data. So if your DNA was pure and of high quality, you'll get a high quality data. So how do you assess that the quality of the DNA is good or bad? So there are two ways mostly. So previously, people used to use spectrophotometers, where they will, see how much light is absorbed by the sample, and based on that, it will quantify. But the problem was that even RNA will absorb some light. Rest of the impurities will also absorb some light in that sample, the buffer. Also, degraded DNA, single-stranded DNA will also absorb some light. But those things do not go into the next steps of DNA sequencing. So quantifying them will actually misguide us and tell us a wrong value of DNA amount that is present in a sample. So for quantification, we recommend the fluorometric methods instead of spectrophotometric method. So fluorometric methods include a dye that will bind only to double-stranded DNA. So that will accurately quantify double-stranded DNA that is present in a sample. So because we are doing a sensitive assay like next generation sequencing, we need to be accurate in quantifying the DNA. Now, once you have extracted DNA, you have purified DNA, you have quantified DNA. These are the three steps you have done, right? So after that, this DNA is now, it is not ready still for sequencing. The DNA that you extract from blood cannot directly go into a sequencer and start sequencing, because what is a sequencer? Sequencer is a machine. And when I give presentations about the products that Illumina sells, Illumina is, just to introduce yourself, you guys, that is the market leader of next generation sequencing. Almost 90% of the genomic data in the world is produced on Illumina sequencers. So there are a couple of other companies that you can study for next generation sequencing, like Oxford Nanopore is there. There is MGI. There are Thermo also has some instruments. So there are different companies that manufacture the next generation sequencers. But Illumina being the market leader, I think if you understand Illumina's technology, then it should be easier for you to get onto the other technologies as well. So we were on the quantification of DNA. Once you have quantified that, DNA is still not in the readable or the sequenceable form. So you need to convert it to in a form that a machine could read that DNA. And that conversion actually is known as library preparation. So this is to be more specific, it's DNA library preparation. So in DNA library preparation, what they do, so for example, this is your DNA fragment, the one in the center, the gray one, okay? and then on the sides you have adapter sequences. So you need to add these adapter sequences to every fragment of the DNA that is in the sample tube. Why we need to add these adapter sequences? There are two reasons. One is the one in purple actually is complementary to the flow cell in the machine on which the sequencing will happen. So this sample will go and bind onto the flow cell because this purple or the blue region is complementary to the flow cell. So this region will go and hybridize there, and then the sequencing can happen of the rest of the fragment. Now, second thing is the purple one. The blue one is the adapter sequence, which is complementary to flow cell sequence. Then comes your purple one. So the purple one is your index sequence. Index sequence is that, for example, if you have 96 samples in a PCR plate, so you will add 96 indexes. You can consider them like a primer for a PCR. Consider similar to that. So it's a similar sort of thing. Sometimes it's 6 bases, sometimes it's 8 bases, sometimes it's 10 bases. Up to 10 bases you have indexes. So indexes are attached with unique sequence in each sample tube. So sample number 1, all the fragments of DNA will have same index. Okay. Sample number 2, all the fragments of that sample number 2 will have a unique index, okay, which is different from the sample number 1. So what happens at the end? All the 96 samples are put in one single tube, because we know that the sample number 1 fragments have which index sequence. So after the data analysis, we can differentiate between the reads that are coming from the sequencing reads that are coming from sample number 1 or sample number 2. So coming back to library preparation, so what do we do in library preparation? We have first of all, we need to fragment the DNA. Okay, so this fragment of DNA that you are seeing in the library, this is also achieved by fragmentation of DNA. Fragmentation of DNA could be achieved by two processes. One is physical fragmentation. For example, if you hammer something, it will break, right? So there is a process called sonication. Sonication is introduction of high sound waves to the DNA fragment, which forms bubble, and when the bubble blasts, the DNA breaks. So this sort of fragmentation, the physical fragmentation, as you can assume, is random in nature, because whatever sequence is in between the bubble where it will blast, that portion will break. Then you have also enzymatic methods to do the fragmentation of DNA. So enzymatic methods, because enzymes are, you can say, kind of like us. They're lazy. They will try to break the DNA where it's easier. They will not go into the difficult regions to break, but it's still, there are very efficient enzymes that are still used in sequencing very commonly, because for sonication, you need an additional, if you are a lab manager, you also have to think about the cost. So, if you want to break by sonication, you need a sonicator, right? But if you want to break by enzyme, you actually need nothing. You will just pipette the enzyme in the PCR plate and it will digest the DNA fragment. So you have to calculate what is what is needed, because if people want to do very accurate, high coverage whole genome sequencing, they might go for physical fragmentation. Some applications need sonication to be more accurate, but most of the applications can give results even by enzymatic. So don't think about good or bad, that which one is good or which one is bad. It depends on the situation and conditions. So you need to evaluate based on that what is worth for you. Then after fragmentation, uh, the library preparation needs to add these adapters. These adapters are ligated by two ways. Adapters can be ligated by ligation using a ligase enzyme, or by doing less cycle PCR. These can bind as a PCR primers, and you can add adapters. So there are two ways of sonication or fragmentation of DNA. I told you, sonication and enzymatic method. There are two ways of ligating the adapters. One is using the ligase enzyme, and other one is by doing amplification of PCR. Once the adapters are added, you will just purify using magnetic beads. So if you guys are interested, I would say suggest how the read how magnetic beads work. You can use magnetic beads and change the efficiency of these beads by changing the buffer solution. So initially, the DNA will bind to the beads, the magnetic beads, and you can throw the supernatant. And when you add elution buffer, finally the DNA will have more affinity for the elution buffer than the magnetic beads, and it will come in the solution. And then you can remove the supernatant and keep it for your next process. After library preparation, because you have fragmented DNA, whether you have done fragmentation by sonication, whether you have done by enzyme, you will know what is the expected range of fragment size for your assay. Also, then you have adapted some known sequences like adapter and indexes and sequencing primer binding site. So these also will tell you the idea that what should be the fragment size. So there is another QC that you guys might have not heard about is the bioanalyzer. You know how much quantity of DNA is there that you have done, but do you know how much quantity is there for 300 basis fragments? How much quantity is for 400 basis sequence? How much is for 500 basis of sequence? Because based on that, it will bind onto the flow cell. Smaller fragments easily hybridize to the complementary sequence because longer fragments, as you can assume, will be lazier. So longer fragments, they need more energy to hybridize onto the flow cell for sequencing. So you also need to do quantification again after library preparation. So do you understand? There is one quality check after extraction and purification, and another quality check will come after library preparation. And here we also want to know the size of the fragment, which we can do by the bioanalyzer. Bioanalyzer is a much more complex form of gel electrophoresis, where you can see that smaller fragments move faster, and longer fragments moves slowly. So it is a more advanced or complex way of gel electrophoresis. You can study the details on internet. It is from, I think, Agilent, the bioanalyzer. Okay, then comes your sequencing. So, how many of you have heard about Sanger sequencing? Can anyone explain Sanger sequencing? Whatever you know about Sanger sequencing. Anyone? Masters in biotechnology, guys. No one?
So basically, we use a set of the TNTPs and dideoxynucleotide triphosphates, which go and bind in place of a DNTP, and it terminates the growth or polymerization of that chain, and we can then conquer the sequence of that particular nucleotide sequence of that particular DNA strand.
Okay, and do you also know about next generation sequence?
NGS. Do you know about NGS also?
Okay, your answer was very comprehensive. I really appreciate, and it was correct and perfect. So it uses DD and TPs, which will stop the polymerase activity, and the next base will not be added because dideoxynucleotide will stop the will stop the polymerase activity, because polymerase is active only with DNTPs. So this sequencing technology, which was Sanger sequencing, in other words, you can say it was sequencing by termination. Okay, so it used to terminate the chain. Next generation sequencing, especially for Illumina, is actually sequencing by synthesis. So it does not, it still uses the fluorescent labeled dyes, but it does not use the DDNTP, it uses normal DNTP. And after addition of each base, the fluorescent signal is captured and different fluorescence is given by different bases. Like A will give a wavelength of some particular value, T will give a different value, then C and then G. And if it's RNA, then we'll talk about U as well. But in general, so we use Illumina used to use 4 fluorescent dyes to differentiate 4 bases. But these days, actually, Illumina uses only 2 colors to identify 4 bases, and it is done by 2-channel chemistry because there are 2 different cameras. One works in the red laser, another one in the green laser. So I'll not go into the much detail how the machine works, but you can identify 4 bases by 2 colors as well. For example, if the red, so C is captured in red channel, okay, 1 wavelength. Then G T is captured in green channel. A will give image in both the channels, and G will not give image in any of the channels. So even with 2 colors, you can identify 4 bases, and this is what we commonly do in our instruments. But in short, aim for this is that you capture the fluorescent signal after addition of bases, okay, and those bases give a signal of a particular wavelength. Based on that, the system will decide that this base is present at this position. So when we talk about the Illumina's sequencing technology, it has a lot of steps. For example, it does something called cluster generation or cluster amplification. So on the flow cell, when the DNA fragment binds, it will hybridize to the flow cell, and it will also amplify on the flow cell. Because if I sequence one fragment at a time of its own type, I will know the sequence and the fluorescence will come only from one fragment. If you get fluorescence only from one fragment, the fluorescence intensity will be very less, and you need a very expensive camera to capture that fluorescence from one single fragment. But and that will make the cost of the instrument very high or unaffordable. So what we do on the flow cell, when the fragment of DNA comes and bind onto the flow cell, because it has complementary adapter sequences, so it will hybridize on the flow cell. So what we do, we amplify 1000 folds at least on the flow cell each fragment. So now we will have fragments with similar sequence, like it will be a cluster of 1000 fragments, and when you will add DNTP to all those 1000, they all will have same sequence because they are amplified from the same initial fragment. So now the fluorescent signals will be 1000 times amplified, and you can use a much lower quality of camera or the cost-effective camera that can give you accurate results. So this reduces the cost of the instruments. So these are the different aspects of next generation sequencing. Then comes your data analysis. So data analysis, so when I say what are these sequencing machines, actually these are the most expensive cameras that you can ever buy. So we have instruments up to $1.5 million cost, okay, US dollars. $1.5 million one instrument. So what does this instrument do? It just takes the images. So it's a camera. So the output of a sequencer is your intensity files. Now converting those intensity images or photos into a base call or a BCL file, which you can see is written here. Okay, so taking an image of the flow cell and converting it to the BCL file is your primary data analysis. Okay, so this is done by the instrument. as well. So those softwares are already on the instrument that you will get the BCL files. This is your primary data analysis, converting, and this primary data analysis sounds easy, converting images into base calls just by studying the wavelength. No, it doesn't work that way. You have to do a lot of background correction, normalization of intensities, offset correction. So one of the experiments that I can do is, if you have any dot in front of you, what you can do is you can point a finger to that dot and then try to point the finger by closing one of your eyes. I don't know, just target any object in front of you, okay, and point a finger towards it and then close one of your eyes and then change the eye. You will see that the finger that you're pointing actually doesn't point at that particular point if we change the eyes. Same thing happens with multiple camera systems in a machine as well. So that is known as offset correction. You need to do the correction of the location of that cluster by doing those normalized. So it is also a complex process, and you need a lot of bioinformatics support to convert that image into a base call file. So these base call files, we still don't know are coming from which sample. So these are all the reads from 96 samples that we took initially, and now we have reads from each of the fragment. Now each of these fragments, when they are demultiplexed, or when the system reads the basis of the indexes, and then bioinformatically with bioinformatics pipeline, it separates the samples. This is known as FASTQ generation. So that is known as the FASTQ file when the sample is differentiated from each other. Now these FASTQ files will then be aligned to a reference genome. So if it's a human sample, we will align this sample to a human reference, okay? And this human reference genome will tell us that this sample which you have tested or sequenced, how it is variable from the reference genome. So that variation could be single base, which is known as SNP or single nucleotide polymorphism, or it could be more than one basis, which can be considered as a structure variation. So these variants, all the variant information uh, is captured in VCF file or a variant call file, which is here. So if you understand what is the flow of the analysis, what we had initially, we had image, then we had BCL file. BCL file, when the samples were differentiated from each other, we got FASTQ. After FASTQ, we aligned it to a reference genome. These reference genome, we see where it is varying from the reference genome, and we got the BCF file. Now we have the variation as well. Now all variations are not clinically significant. Maybe there are hundreds of variations in your genome from the other genome, from a reference genome, but they do not have any effect on the phenotype. So some variants have clinical significance that could be associated to a disease, which could be carcinogenic, or it is diabetic prone, or any disease. There are multiple factors that we can talk about, but it should have some clinical significance. Now, how we can know what is the clinical significance? There are databases, like you might have heard about one of the very common one, which is known as ClinVar. Okay, so ClinVar DB, if you will Google, you will get to know about this database. It has a lot of variants and information about the diseases that these variants are associated to. So there are multiple databases of such types. So all of these variants are then matched or aligned with the databases, annotation databases, and now this annotated data finally will give you the variants that are clinically significant. So this is known as tertiary analysis. So first, converting image into the base call is your primary analysis, okay, and then getting the sample FASTQ files is also your primary analysis. Then aligning these FASTQ files to get the BCF file is your secondary analysis. Then annotation of BCF file to get the clinical significance is your tertiary analysis. Okay, so that was it. I did not prepare in a presentation form because I wanted to have the flow. It was very much, I think, complete and comprehensive. This is just the tip of the iceberg that I've talked about. Each of these topics, actually, I can speak for like 5 hours on each of them. And I have slides of 80, 90 slides at least for each topic. But now, what I want to know is what questions does it put in your mind? I was still not focused on the technical part. I want to know what do you think about after hearing half an hour me talking about next topic. But now, what I want to know is what questions does it put in your mind? I was still not focused on the technical part. I want to know what do you think about after hearing half an hour me talking about next generation sequencing?
Any questions come into your mind?
What is the first thought, first question? Anything. It could be technical, it could be career perspective, it could be any random question. There is no wrong question. Ask anything. Yeah. Okay. So let's talk about now the career perspective of it. So when you do biotechnology, like we took biology in 12th, we knew, our parents told us that you need to be a doctor, take biology. Most of us, right? So this is what we think. When we do biotechnology, the problem is we have this limited exposure. At least in my era, it was there. Maybe you guys have more exposure now. But what we understood at the time when we were doing bachelor's, that we have to do MSc, then we do PhD, then we do postdoc, then we do another postdoc, then we do find some job in a university or something. So this is what we know. But actually, there are a couple of things that we can do after biotechnology. First, of course, is this BSc, MSc, PhD approach, which can take you which can take you to a research institute or to a university. Simple. This one most of you know, and which is where you have most common people available because this is all what they know. But to my understanding and my personal experience is that you have a better option, okay, which I mean, if research doesn't interest, if research is your passion, I would not stop you doing it. Okay, if research is your passion, go for masters, go for PhD, and go for your university or academic career. But I don't know if that's your passion, what's passionate about it, because there are many happening things that you can do outside this career. So what I can tell you is, one is the food industry that the guy highlighted. Another is hospitals have hospitals and diagnostic labs can also are actually playing the applied science part of this life science. Okay, so these genetic courses, genetic testings are not very common still in India, but there are actually major part of the Western world and becoming very common in Middle East. And also we have now even the Modi government has started the genome project in India. So 10,000 genomes. The Karkinos Institute in India, there are two, I think, branches who have got Illumina sequencers, the same instruments, 1.5 million. So they have got like 2 or 3 instruments recently this year. So that genome project is also kicked off. So India is also now becoming a hub. NIPT is one of the most common tests that is done for pregnant women to test the number of chromosomes. You might have already studied Down syndrome and Turner syndrome, trisomy 21, 18, 13. All of these actually are also tested more efficiently on a sequencer and actually on Illumina sequencer. So these are different perspectives. So in the job perspective, what you should do, so there are two types of job. One is you are inside a lab, you are buying the technology. The person who's selling you the technology actually is also a scientist, because I need to talk to a doctor, I need to talk to a research academic person, I need to talk to a clinician or a technician in the lab. So all of those people who are selling the science are also scientists, the technology. So there are two perspectives. One is on this side of the table and the other one on the other side of the table. So you can choose now. Lab technician, lab manager, lab technologist, these I think are pretty much clear. That exposure also you will get from this lab where you are sitting, and it's a very good hub, and they're doing a broad spectrum of tests. So one aspect is this, that you can get. First one I told you, the research one, the normal, which all people know, the academic career. Second is you are in a lab, whether it's a diagnostic lab or it's a research lab, you are buying the technology. The last one that I want to focus is right now is selling the technology. Now, selling the technology will have couple of options. One is you can be in the sales for that particular. Now, we think sales and marketing are not a good job in India, but trust me, if you will join any company, only sales and marketing people rule. Rest everyone is labor. So, so sales and marketing, first of all, gives you exposure to talk to a new person. That gives you confidence. It enlightens actually and brightens and sharpens your personality. Okay, so talking to new person. So what is the most difficult task in this world? To get money out of someone's pocket. Nobody will give you money, except your parents, of course. I'm not talking about that. But to get money out of someone's pocket is the most difficult job in this world. And that is what salespeople achieve. So, how do they achieve it? I mean, they have some sort of skills, and don't you want yourself also to have those skills? So, don't think about only in one direction: BSc, MSc, PhD, postdoc. I'm not saying don't do it. If you're passionate about it, do it. But if you are not passionate about it, it's not the end of the world. There are 10 more better options which you can excel in to explore. So one is in the diagnostic labs, you can gain experience, and also to be specific to your question, you ask how to go to the Middle East or all of these countries, or to go abroad for these things. First, you should have a diagnostic lab experience in India. Also, you should have license qualified for that particular country. So, for example, I know a little bit about the process for Dubai. So, there is something called DHA, that's Dubai Health Authority, and there is entrance exams. So if you guys have done entrance exams for BSc, MSc and other things in India, it will be very easy for you. It's not a difficult one. So that is kind of a formality. But main thing is that you should have your documentation correct. When I said, be prepared and outsmart others by being prepared, actually one of the things was your documentation should also be very strong. People actually do not know the documentation process, and that makes the process difficult. But otherwise, coming out of India and having job options is easier if your documentation is strong. You should have all your degrees attested by the HRD ministry and the embassy, whichever country you want to go in. You should have a license, good standing certificate from the lab. So good standing certificate is very important if you want to go out. Yeah. The other option that I said, selling science and technology, it has two options. One is you can be in sales, and the other thing, you can be in support. So if you want to be very even in commercial aspect, you can come into a position like I am in. I am a field application scientist. So my role comes in when the product is sold. So I will give training to all these labs how to set up the experiment. And if they face any difficulty, I will troubleshoot the problem scientifically. There might be issue with the quantification or the library preparation. We'll evaluate the data and help them in project designing. So all those scientific discussions are done by post-sales support. One is sales or pre-sales. Pre-sales support is marketing. So marketing is also very technical. You lines in your country or in your region, and then you can suggest to the company, this product should be good in this region, we should spend more on R&D for this particular product. So I think we have hit the time. So any more questions, I'm happy to answer. Otherwise, we can also take and note down questions, and I can respond them later. So I'll give you 5 more minutes to ask questions.
Hi, sir. So by God's grace, we have well-established business. So sir, I wanted to incorporate this biotechnology in that business. So what are the options available?
What business is that? You have to tell the business concept.
So yes, but I want to start with small scale business for 1 billion from now.
I mean, it's a broad spectrum of life science industry. You can go into the food testing, you can do the water resource management things, you can do NIPT, for example, that is for clinical testing. You can do something like what the setup we have for Shahbaz Lab, the PCR, qPCR, and all these diagnostic tests, CBC. I mean, it's a broad spectrum. It depends how much you want to put in. I mean, if you want to put more money, you will have less competition because, of course, the smaller labs you will open, there are more number of smaller labs. So it's a very broad spectrum. I mean, I cannot tell you one single thing that you can do this because it could be. I am from genomics, so I can tell you genomics, but that's not the end. Maybe we can get more money in food processing or in water resource management and some other agricultural applications. Any more questions?
I think there are no more questions for us. They are done.
Position and time, guys, and I wish you all the best for your careers and future. And in case you need any help, Shabazz can share the LinkedIn details, and you guys can text me there.
Okay. Okay. Thank you so much, Faraz. It was a wonderful session, and I hope